Okay, hello everybody. We are going to continue here. We're on week 4 now, and if we go into Blackboard and we look at our weekly folders, and in week 4, well, what does it say? It says in this week's folder, you'll find 3 things: the lecture video for Chapter 3, which is textbook Chapter 3, a lab video that we're going to do. We're doing that right now. And then a supplemental video that shows the anatomy of the fetal pig. All right. So when we open that up, you'll see those things. And there's just a reminder to look at the schedule. Make sure you have the schedule handy and you're looking at it, because next week we're going to have 3 assignments that will be due in addition to 1 lecture. These assignments are going to be the lecture exam, 1, the lab exam, and the disorder project, which I will explain how to do that next week. I'll supply the rubric for that. All right, so let's look at this lab. It's lab 4, and normally we would come in and we would have pigs. You and your lab partners would grab 1 pig, put it in the dissecting tray, and then you would get started. Obviously, we're not going to be able to do that, but we could kind of do this electronically. All right, so let's kind of jump ahead here. This is just a diagram that shows you the different types of cuts that you would be making. It recommends that you tie the limbs back. These are the tools that you would be using. If you wanted another visual aid in addition to the ones that I'm providing you with, and there's a link here that the college recommends. And then there's safety warnings. Of course, wear gloves, clean everything after you're done with it. It just shows you what to do with the parts. After you're done, you would put it back in the plastic bag. Any loose parts would go in a bucket that we have in the lab. And then it would kind of instruct you on how to identify the gender of the pig, all right, because both males and females have rows of nipples and an umbilical cord. However, the male's urogenital opening would be near the umbilical cord, and the females, they also have a urogenital opening. However, it would be closer back towards the anus. So if you look under the tail at the anus, if there's 2 holes, you have a female. If there's only 1 hole back there, you would have a male, because the second urogenital opening for the male is up along its belly underneath the umbilical cord. So you would probably be doing that first, and then you would get your pig on its back, start cutting it open, being careful not to cut into any internal structures as you try to open up the different cavities. So this is just a list of structures that the college is asking you to find. All right, so we'll go through this list, and the first thing that you would probably do is you would expose the thoracic cavity. So you would probably start opening up in the neck region through the pharynx and exposing all of this below. This area right here to about here, this is called the thoracic cavity. And what structures do we have in here? Let's see what we got. Well, this structure right here is the voice box. And we call it a larynx. So when you breathe in through the nostrils, it goes through the pharynx and then through the larynx and down this tube. This is the windpipe here, but we don't call it a windpipe. It's actually called a trachea. This structure is very easy to identify because there's cartilage rings that are embedded in here. These are called cricoid cartilage rings. And these rings are important because they hold that tube open all the time. If you were to pull this to the side, push it to the side, there's another tube behind there. And I'm talking about this one here. And this is for food. This tube would be the esophagus. All right. This structure right here is the heart. And you could see we have two chambers that sit up on top. This is the right atrium. This is the left atrium. These structures are also sometimes referred to as the auricles. But we're just going to label it, the whole thing as a heart. Sitting on top of the heart is a gland. This is the thymus gland. We have two lungs. There's a lung here. And here's the lung here. A couple different lobes. This is the right lung. And this is the left lung. And then we have this big sheet of muscle here that separates this whole cavity. Remember, I told you this whole cavity that houses these organs here, this whole space, it's protected by the ribs. You can see the ribs in here. This whole cavity, this is called the thoracic cavity. The thoracic cavity has a wall here that separates it from the next cavity below. The next cavity below, down here, this would be the abdominal cavity. The abdominal cavity houses all the digestive organs. So we would find the stomach, the liver, the intestines, all that stuff down there. But anyway, this sheet of muscle here, this is called the diaphragm. This is the muscle that controls your breathing. All right. So now we're looking inside the abdominal cavity. So the diaphragm would be up here. All the organs that we just labeled would be above, up here in the thoracic cavity. So now we're looking inferior to the diaphragm. We're looking at the whole cavity here. This is called the abdominal cavity. Abdominal cavity. All right. This big structure here, this is the liver. Oh, put that in the wrong spot. Liver. What this arrow is pointing to here is this little pouch underneath the liver. See, one of the functions of the liver is to produce bile. Bile is a chemical that helps to break down fats. And when the bile gets produced in excess, it gets stored in this structure, which is kind of just like a compartment of the liver. This is called the gallbladder. All right. This structure here is a big sac. Very easy to find. This is the stomach. There would not be any food in there because these are fetal pigs that haven't eaten anything yet. You'd be able to peel off this very thin structures. It almost looks like a tongue. And it's held on by this light tissue here to this deep area here. This is called the spleen. We do have intestines in here. This whole squiggly bag of intestines right here is called the small intestines. And then this larger collection here, it would look more green. These are the large intestines. All right. On both sides, they're only showing one, but on both sides, if you pull the digestive organs out and push them to the side, deep to the intestines, you would find kidney. The kidneys have tubes that extend down from them, and they lead to the bladder. The urinary bladder would be this structure here. These tubes are called the ureters. And we forgot about this one. If you pull the stomach up and the intestines to the side, there's a very faint organ in here. It almost has a little yellowish tint to it. And you could see it has some texture to it as well. This is the pancreas. All right. The video that I show you is a female pig. So kind of near the bladder, you would see these squiggly lines. It kind of looks like noodles. And at one end of the line of those squiggly tubes, there's ovaries. And at the other end of both of them, they lead down to the uterus. This is showing you the testes here. So this is a male pig. You would be able to pull this urinary bladder. This is the urinary bladder here. And you would see these tubes, these two sets of tubes. I don't know. This picture is very difficult to see. But there would be like little sacs on one end. Those would be the ovaries. And if you followed them deep down, you would see in here would lead to a uterus down in there. This picture doesn't really do it much justice. All right, so inside the mouth, well, we would have the tongue. You would have to cut the jawline back to see these structures here, but there's a little flap of tissue here that covers the trachea. So when this animal's eating food, the food gets diverted away from the trachea and pushed further back towards the esophagus by this structure right here. This is called the epiglottis. So the epiglottis is important because it prevents from choking. If you were to rub your finger on the roof of the mouth, it would feel very rough here, and it would feel very smooth here. This has a very rough texture to it. This is called the hard palate. And this is called the soft palate. All right, so what we really are interested in is taking what we learned from the pig and kind of translating it over to the human. Remember, this class isn't really called pig biology, it's called human biology. So we would have these models in our classrooms, and it's important to be able to translate what you see in the fetal pig onto the models. So if we look here, this is in the neck region. That's the larynx. This gland right here is also in the neck region, just inferior to the larynx. This is called the thyroid. The thyroid is a gland that it produces hormones that regulate your metabolism. As we go down further, we have the heart here, surrounded by lungs. This is the left lung. This sheet of muscle here is what separates the thoracic cavity from the abdominal cavity. This is the diaphragm. Here is the liver. And And underneath the liver, if we were to look under there, we would see this sac. It is greenish. That's the gallbladder. The gallbladder stores excess bile. Underneath the liver, we have the stomach. This is the large intestines. And this is the small intestine. So a little bit different when we compare it to the pig, right? The pig had a package of small intestines wrapped up in a thin tissue called mesentery, but the large intestines did not surround that collection of small intestines like it does in a human. In where the small intestines empties into the large intestines, that area of the small intestines is called the ileum, right? So the ileum drains the food nutrients into the large intestines, right? Specifically at the part of the colon here, when the large intestines is called the colon, that would go up. So this part of the colon is called the ascending colon. It goes up, ascending colon. And then from here, it would go over, across. That's called the transverse colon. And then it would go down in the descending colon. And then it would make kind of like an S-shaped turn. That's called the sigmoid colon before the food material would be stored as feces in the rectum. So here we see the ascending colon, the transverse colon, the descending colon. We don't really see the sigmoid colon, and we do see the appendix here. Bad color choice for this appendix. The appendix is, so this is the ileum, which is the last part of the small intestines draining into the ascending colon. So it'd be down here. And we have this structure here, which is the appendix that houses a lot of bacteria. All right, so if we take the heart out, we have a notch, right? We have a cavity. This is called the pericardial cavity. You can see the left lung has a little notch in it. All right, because it has to make space. But we have some tubes back here, right? So this is the trachea. This is the windpipe, this white one here. And that trachea splits into two, right? This is the bronchial trunk here. So this is going to split into the right bronchus and the left bronchus. And that's what brings the air deeper into the lungs. All right, so we have up here, it's still the trachea. So we're going to label that trachea. This is the esophagus. And this is the right lung. This big sheet of muscle in here is the diaphragm. This is what our spleen looks like. It's also on the left side, just like the pig. Then we have a kidney. Sitting on top of a kidney, we have adrenal glands. From the kidney, there are tubes that deliver urine from the kidney down to the urinary bladder. These are called ureters. And then this structure here is the urinary bladder. All right. So what I would like you to do is go back, look at the cavities. The dorsal cavity includes the cranial cavity and your spinal cord area, right? So what organs will we find in these two cavities? The ventral cavity is your front, right? So when we look at the ventral cavity of the pig, we had two major cavities, the thoracic cavity and the abdominal cavity. When we include the reproductive organs, we refer to it as we go down further into the pelvic region, the abdominopelvic region. But there's really two different cavities. So what organs are pleural? Which ones are pericardial? What organs do we find in the abdominal cavity? Both of these are thoracic. The abdominal cavity, and then what organs do we find in the pelvic cavity? And then which structure separates the thoracic cavity from the abdominal cavity? We mentioned that a couple times. All right. All right. So what I want to do here is just talk about very briefly. Get my picture out of the way. I gotta. What's going on inside the small intestines? If you think about the small intestines, and you think about it like a tube, right? Let's see here. Let's draw this like this. Imagine we have this tube going, and let's say we ate some food, and now we have some food nutrients traveling along the tube. Nutrients that we want to get sent into our blood, right? So, of course, in the intestines, there's a lot of blood. There's a lot of blood vessels down in there because this is where the blood goes and picks up nutrients. We need nutrients in our blood, right? That's an important part of, making energy and whatnot, right? So you got a lot of blood vessels down there. So the idea is, how do we get these food nutrients from the intestines into the blood? Well, it's not a smooth surface like this inside the intestines. It's there are folded structures that we see in there that are almost like these finger-like projections that go up and down all along the inside surface of the intestines. And what this does is it's creating more surface area. These structures, by the way, these finger-like projections, these are called villi. And what they do is they provide more surface area for the absorption of nutrients. Which is kind of important. So if we were to look really closely and zoom in on one of these villi, well, what would it look like? Well, we would see two types of cells. We would see, number one, these structures called goblet cells, and then we would see columnar epithelial cells, all right, inside the villi. Now, columnar cells, if we look at columnar cells under the microscope, they call them columnar cells because they look like columns. They're arranged like little columns. So they're kind of long and skinny like this. They'd have a nucleus, and they are arranged next to each other like this. So this is called, because there's only a single row of them, this is a simple arrangement, simple columnar epithelia cells. One unique feature that columnar cells have are these little microscopic hairs that kind of go along the top edge. All right, we refer to those as microvilli, and they even further increase the surface area for absorbing nutrients. All along the villi, we would have these other specialized cells embedded in between here. And these cells are called goblet cells. And they actually would stain. If we're looking at this slide, they would actually be more purple. They stain purple. But they would be embedded in the middle of these columnar cells. And then the columnar cells would continue like this on both sides. So what do these columnar epithelial cells do? Well, they're actually absorbing nutrients. So as nutrients are passing by, they're absorbing them. And what do these goblet cells do? Well, they do one thing. They produce mucus. So if you were to rub your finger inside your intestines, it would be very slimy and slippery. And that's because of all these goblet cells that are in there. So what important process following digestion, is facilitated by the villi? Well, the villi are packed with these columnar epithelial cells. We would say absorption. Absorption of nutrients from the intestines into the blood. And what is the function of goblet cells? Well, they secrete mucus. So where the villi are, here we see a villus. Here's another villus. Here's another villus. Collectively, we call this the villi. This is a villus. We're going to label just this one. This whole area that contains all the villi, because there would be goblet cells going around next to the epithelial cells, this is called the mucosal layer. The layer beneath it that contains the blood vessels is the submucosal layer. Deep to that, or underneath that, we would have all this smooth muscle tissue in there. That's called the muscularis. All right. And then you would have your microscopic observation. So let's go to the lab atlas and see what it is that we need to look at. Now, the lab atlas is a great tool because it summarizes everything that's in the lab, right? So if we go here, we would see this is the microscopic image. This is using scanning lens, right? Scanning lens. So this is not very zoomed in, but we could see the layers here. We see clearly we could see villi running up and down. All these dark circles that are on the edge of the villi, those are the goblet cells. The lighter regions in between would be the columnar epithelial cells. So we see everywhere that we have villi, that's the mucosal layer. Below that is the submucosal layer, so that would be packed with blood vessels. And then where we see all this smooth muscle is the muscularis. If we were to zoom in, now we could see, this is high power view. These are where the columnar epithelial cells are. Let me move my picture out of the way. And these that I told you before usually dye purple. Those are the cells that are making mucus. Those are the goblet cells, all right? So this is the drawing that you're going to be doing, and this is the high power view. So the field of view is 450 micrometers. So what does that mean? That means from one end all the way to the other end, the distance is 450 micrometers. Well, what would the width of one villus be if the entire distance across the field of view was 450 micrometers? What would it be? Well, let's go back here. So you're going to draw that. In here, be sure to identify the mucosal layer. So everywhere that there's villi, draw and label the goblet cells, the columnar epithelial, and try to get some of the smooth muscle layer in there. Remember, I want this drawing to be on high power. Does that say that up here? Yeah, it says it right here. So when we're using high power, remember the eyepiece is 10x, the objective lens is 40x, total magnification is 400x. Keep in mind the field diameter. What does that mean? That means the distance from here all the way across to here is 450 micrometers. What I want you to do when you draw your villi in the notes section, I want you to estimate the width of one villus, right? And of course, I want you to do that using the units micrometers. So what you do is you would ask yourself, let's say I drew a villus, right? Let's say I drew something that was this thick. I'm not going to draw a villus. I'm just going to draw a rectangle. And if my teacher is asking me to estimate the thickness or the width of this rectangle, right? First thing I would do is I would ask myself, well, how many of these things can I fit going all the way across? And it's just an estimate, right? So I would predict I could do like maybe 1 across, 2, 3, 4. Let's say I say 5. Well, if I think I could fit 5 of these going all the way across, well, then the estimated width would be 450 divided by 5. 450 divided by 5 would be 90 micrometers. That would be the width of this. But I'm not drawing a rectangle. We're drawing villi, right? So use the lab atlas, these images here, to draw your high power view. Okay. And then estimate the width going across and draw this to scale. All right. And I think that is it. All right. So good luck with that. If you have any questions, give me an email. But I think it's pretty straightforward. I mean, for the lab exam, I'm just going to be pointing to pigs' parts, and you are going to be responsible for just identifying them. Also parts in the human model, right? As far as this lab is concerned. All right. Okay. Take it easy.